Welcome and hello, everybody, dialing in from all over the world to this Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're really glad you're making the time to dial in today. We will be talking about sequence-selective translation inhibitors with Larry Hammond today. Good morning, Larry. Good to have you.
Good morning. Thank you, Dennis. Glad to be here.
And now, without further ado, I'd like to introduce our guest, Larry. Larry Hammond is the co-founder, president, and CEO of Interdict Bio, pioneering small molecule context-dependent translation inhibitors for addressing historically undruggable targets in oncology and neurodegeneration. Previously, he was global head of discovery at Takeda, and prior to Takeda, he was corporate VP and global head of small molecule drug discovery at Celgene. Prior to that, he had held positions of increasing responsibility at Novartis, Bristol Myers Squibb, and Ligand. In his career and more than 30 years in drug discovery, his teams have advanced over 22 molecules into the clinic, including saxagliptin for type 2 diabetes, the clastasvir for HCV, and the mRNA splicing modulator branaplam for SMA. At Celgene, his teams built a state-of-the-art platform for molecular glue and heterobifunctional protein degradation, therapeutic discovery, and development, with several molecules from this effort, including mezigdomide, golgadomide, and gringdegalutamide. Those are all targeted protein degraders that are currently in phase 3 for hematological and solid tumor oncology indications. Larry is really prolific and a co-inventor on over 70 patents and co-author on more than 90 scientific publications. He served as a standing member of the NIH SBCB study section, and also at the moment helps several academic boards as well as biotech boards as well. Larry is highly decorated with awards. A few of those include the ACS Heroes in Chemistry Award, the ACS Division of Medicinal Chemistry Award, and Larry was also inducted into the Medi Hall of Fame as well. And yeah, Larry got his PhD in organic chemistry from the University of Michigan and overlapped with our very own Lou Pennington there. So welcome, Larry. It's a pleasure to have you here today.
Thank you, Dennis. That was a very kind introduction, and I very appreciate it. Really glad to be here. I love the Drug Hunter Flash Talk series. So,
Awesome. Yeah, thank you so much. Yeah, Larry, and in the good old tradition at Drug Hunter here, I'd want to ask you some personal question. Can you talk a little bit about what inspired you to start a career in drug discovery?
yeah, thanks. So I think, like many of us who have trained as a synthetic organic chemistry in graduate school, where you're deeply immersed in how to synthesize molecules that possess some interesting activity, you are drawn to, continue learning more about the relationship between structure and function, et cetera. And the logical path then, I think, is to pursue medicinal chemistry, where you can really broaden the scope of your thinking about the problem from not just how to make the molecules, but how that relates to their biological activity, et cetera. And I think that's it's just a fascinating journey of learning that never stops.
Yeah, totally agree. I'm also super fascinated by getting the data, always then improving the molecule activity, ADME properties. That's something that's super fascinating to me also. What's your favorite molecule, Larry, and why?
Oh, that's a very tough one because there's so many wonderful molecules out there. And when I was thinking about this question in advance, I think I would have to say the molecule that became declatosphere, which was the first in class HCV NS5A inhibitor that we worked on at Bristol Myers Squibb and became the drug Declinza. And partly because this was early in the advent of the use of phenotypic screening for discovering novel targets for disease intervention, but also that the molecule was a rule breaker in terms of what was very popular in the early 2000s around rule of five kind of things. And it really was a dogma busting kind of molecule, 750 molecular weight, really looked rather strange and ugly. But the properties and the behavior of the molecule nonetheless were really outstanding for oral delivery. And this really, I think, cemented my love of trying to break dogma in other places and really follow the quantitative science and the laws of physics rather than be bound by artificial rules.
I love that. Yeah, I think it has an acetylene also in the middle, a somewhat symmetric, the molecule,
It's a
right?
symmetric molecule, yeah.
Yeah, it's an amazing, amazing molecule. Also, one of my favorites too, I say. Um, what are you most excited about in drug discovery today, Larry?
Well, I think first of all, there's such a new renaissance and proliferation of small molecule modalities. Of course, my entire career has really been focused on small molecules, and I think those are, ultimately the most convenient for patients and the most broadly accessible to other parts of the world. And I think, when I see molecules that aren't just blocking the function of a protein, but able to induce downstream consequences that are even more powerful, that can work in a substoichiometric manner, I just feel like we're just at the cusp of being able to really address a wider scope of biology with the things that people are coming up with. So I'm excited to be around to see it all.
Awesome. Yeah, and you're at the forefront of it all, actually, I think. That's amazing. If you could go back in your career, Larry, and give yourself some career advice, what would you tell your younger self?
I think just that, yeah, to always stay curious. Don't be trying to do something that's guaranteed success. Always take a little more risk and be a little more it's a lifelong learning endeavor to be in the field that we're in and to continue to stay curious.
Absolutely. Yeah, and last but not least, what do you like most about Drug Hunter, Larry?
Oh, well, there's so many things. I really love the Drug Hunter team have curated such interesting content from across our ecosystem. And of course, the flash talks are always enjoyable, but just the useful information and the perspective from people who have actually spent a fair amount of time in the trenches doing drug discovery and the perspective and the insights that are again put together and curated by your team are just I find to be a rich source of information for people that are either starting out in their career or even for old people like myself that still can learn a lot from what you guys put together.
Awesome. Thank you so much. That means really a lot to the team, especially coming from you, Larry. So thank you so much. And yeah, I wanted to quickly show people one kind of function at Drug Hunter that maybe people can use a little bit more, and that's the structure search. So in the structure search, we actually have three types of databases, and I just want to show maybe how we can search those databases, maybe with some relevant molecules, drawing a little bit of an image here. And yeah, we can see maybe the evolution of how maybe, some of the degraders are maybe approved and were discovered. So we can see what are the FDA approved molecules that contain these types of IMiDs here. And you can see like when they started to get approved, of course, there's a lot of history with thalidomide, for example. But if you would like to see, let's say, more modern molecules, you can also look in our modern clinical compound database, and you'll see the evolution. Now also there will be heterobifunctional degraders, of course, and here you can sort this by date. You can see now in the later years, there's this, of course, this functional group has been used much more in heterobifunctional degraders. So that is really visible here as well in the clinical candidates. And then last but not least, you can kind of go back and also survey the drug hunter molecules. And here we serve up really kind of the latest molecules and discoveries. And really, it brings you to the most relevant compounds. So, as opposed to something like SciFinder, we don't serve every molecule. We just serve essentially the most relevant molecules. And then you can also click in here, for example, if you want to learn more about a specific molecule. We typically have case studies for these drug hunter molecules, and you can learn, why these functional groups were used. We can learn a little bit more about the SAR in that particular series. You can learn more about the target and really the whole discovery program, and even the binding, clinical results initially, and really educate yourself very quickly on these types of projects. And with that, I will stop sharing and hand the screen to Larry. And yeah, we're very much looking forward, Larry, to your presentation about those interdictors.
Great. Yeah. Well, thank you again for the wonderful intro. And it's a pleasure to tell our story of what we're doing at Interdict with our amazing but small team. And again, particularly for this audience, it's a real privilege. So I'll start with somebody who can introduce the overall feeling that we have about how this company is on the cutting edge of things with a short quote here.
If the other thing I would say is that if you feel safe in the area that you're working in, you're not working in the right area. Always go a little further into the water than you feel you're capable of being in. Go a little bit out of your depth. And when you don't feel that your feet are quite touching the bottom, you're just about in the right place to do something exciting.
Well, that kind of sums up what we're pursuing at Interdict. It's again a little bit at the edge of possible, but the team is making it possible every day and continues to blow me away with just discovery after discovery. So to introduce the topic here, interdictors are these oral small molecule sequence selective stallers of translation of mRNA to protein. So they block the synthesis of disease-causing proteins, and they operate inside the large subunit of the ribosome, which I'm showing here in the cyan color. And the thought is that rather than wait till a problematic protein is formed and try to inhibit its function or to induce its degradation, let's stop it from being formed in the first place. And particularly, this is applicable for oncogenic transcription factors, for instance, where even a transient expression of the target could do a lot of damage. And so the name interdict kind of comes from the military usage of the term to meaning to destroy enemy resources before they even get to the battlefield. So because we have a common binding site for every target in the entire proteome, this modality is broadly applicable across all target classes and disease areas, but we've chosen to focus so far in oncology and in neurodegeneration. I'll tell a little bit of a story on each of those topics today. So just to reorient everyone on the basic mechanics of translation, here I'm showing the two subunits of the ribosome reading along a messenger RNA. And now we see the transfer RNAs bringing in each new amino acid to add to that growing chain until you encounter the stop codon. You recruit release factors, and then the peptide is released into solvent where it can perform its proper function. And because the ribosome is energetically costly for the cell to manufacture, it gets recycled and reassembled on the next molecule of mRNA. So now here I'm showing translation as before, but we're going to see an interdictor molecule bind in this pocket in the peptidyl transferase center of the ribosome and then exert selective friction against that peptide chain as it's moving over it. This has caused slowing of the ribosomes and ultimately ribosome collisions. And then quality control mechanisms come in and degrade the mRNA, and the cell gives up the process. Well, there's actually a precedent for this modality in the form of an approved drug that's called Cinribo. It was developed by Cephalon more than 10 years ago. It's a natural product called homoharringtonine, and it's used in second line for CML after failure on Gleevec, et cetera. It's a rather large molecule, so it occludes the entirety of the peptidyl transferase center, and as such, it's not a particularly selective molecule. Nonetheless, it has sufficient safety and efficacy to be FDA approved. But perhaps a more relevant precedent came from my two co-founders at UCSF, Jamie Fraser and Dennis Fujimori, where they investigated an older Pfizer antibiotic molecule called linezolid. So linezolid had historically been thought to be a global inhibitor of translation in the bacterial ribosome, but they showed through biochemistry and cryo-electron microscopy that actually is a sequence-selective inhibitor of the translation. Here, the linezolid molecule in yellow loses its activity when this alanine, for example, in the peptide chain is replaced with a glycine. So this gave us the impetus to pursue this in the context of the human ribosome for therapeutic intervention. So again, when we think about normal translation, we have relative even spacing of ribosomes along a message. That syn-ribo molecule that I mentioned, because it blocks initiation, it blocks the very first peptide bond formation in every context, it's more of an initiation inhibitor. The next closest things would be GSPT1 degraders. So GSPT1 is also known as ERF3, which is the eukaryotic release factor that's required to release translated peptide from the ribosome. And so by blocking GSPT or degrading GSPT1, you prevent the offloading from every ribosome indiscriminately, again, affecting every translation event. Whereas in our context, the molecule is having direct contact with that peptide chain as it's going by and reading the sequence in a way that provides selective stalling. So we didn't have to go far to find chemical starting points that were of interest. Here I'm showing overlays of a number of molecules that have been studied largely by the academic community over the last several decades. And among these are, of course, this homoharringtonine molecule that I spoke about earlier and the linezolid type analogs. But it also includes erythromycin that I'm sure people are more familiar with, and a very compact and very ligand efficient starting point of anisomycin that became of interest to us as a place that we could begin to make modifications to build new chemotypes here. And one of the very first things we did was to take that very simple anisomycin structure. Of course, it has a labile ester moiety, so we can replace that with something more stable like a carbamate, and just put simple derivatives on to that linkage where those moieties project directly towards that peptide chain as it's going over, and show that we could have selectivity for engagement of different sequences. And so we use a technique called ribosome profiling, where you basically are looking at where each translating ribosome is stopped, and that's based on the inclusion of the molecule. And what we can see is when we introduce a basic moiety like this pyrrolidine, we have strong preference for glutamic and aspartic acid, as you might expect. And when we introduce a more lipophilic nonpolar moiety, we have strong preferences for alanine, proline, etc. And this was the basis for the earliest discrimination of different sequences, but we've subsequently built multiple categories of molecules that can have strong stalling preferences for different 4 and 5-mer amino acid groupings, even up to 100-fold discrimination. And this is a paper that we have out on bioRxiv that I'd encourage you to look at for additional details. So, but it makes sense that the composition of amino acids is quite diverse and can be recognized by molecules by the same underlying physical chemical properties and biophysical forces that govern any bimolecular interaction. It's just that in this context, it's a dynamic system where the linear chain is gliding over your molecule, but it's sequential iterative interactions with different targets. And you can see just by looking at these three targets, for example, that there's quite a bit of a different signature that can be read by the molecule. Not to mention when we go into things like neurodegeneration, where you have in some cases, these long stretches of a single amino acid or a duplex that really create different signatures as well. And it's not just the physical chemical properties, but the size as well. And so the steric component is important. Here, I just represent that in a more mathematical way because this is a part of how we can reduce this modality to be amenable to machine learning. Here, we can give a score on different parameters, whether it's by sterics, by charge, or inclusion of prolines, which happen to translate at half of the speed of every other amino acid. So we can say for target A versus target B, there's quite a bit of difference of how a molecule would sense that as this peptide chain is gliding over the molecule in that tunnel. Likewise, where the charge signature that is gliding over, it can be quantified, and we can use this to iterate on our machine learning. And again, the inclusion of prolines are like extra little speed bumps along the way, and their positioning relative to a sequence of interest for intervention can have a compounding effect, which we can capitalize on as well. So, as you might expect, the structural aspect of this is incredibly enabling, and we have to date solved almost 30 high-resolution cryo-electron microscopy structures that really guide our structure-based design with the use of the Schrodinger tools. Here I'm showing two different cryo-electron microscopy structures, one at a time, and I'll walk you through them. So here what I'm showing is to orient you, in blue is the ribosomal RNA, which it comprises the entire architecture of the interior of the ribosome. In green is the transfer RNA, and in salmon color here is a sequence of the oncogenic transcription factor MYC that is the preferred sequence for the maximum friction for the molecule that I'm going to show you. This is EQEDE, so again, rich in glutamic and aspartic acids. And what you're going to see here is this conformation of this peptide chain is, in one sense, in the absence of an interdictor molecule. And with our interdictor molecule, you can see that reorientation of that side chain to make a perfect salt bridge between this pyrrolidine moiety and this aspartic acid side chain. So we can use these dynamic movements that are induced by the presence of our molecule to better understand how we can find and enhance and continue to evolve for even better sequence selectivity as this is moving by. And we use a machine learning platform called the Interdictionary that is kind of putting these pieces together. So it's not only the spatial and binding interactions that come from the cryo-electron microscopy and computational chemistry, but translation kinetic assays, the sequence preferences that we discover by ribosome profiling, and then public databases about sequences of different targets, together with physical chemical properties of our molecules to kind of put these pieces together. And we've been recently partnering with NVIDIA to help make the different pieces of this overall interdictionary talk to one another in a better way. So these interdictors kind of take advantage of many of the best properties of the contemporary small molecule modalities, and I'm showing here three that happen to be things that I've had the privilege to be part of working on in my past. And so, like RNA splicing modulators, because we're going after the target when it's a fraction of the copy number of what it will be as the protein, there's a favorable stoichiometry associated with that. Like molecular glue degraders like mezigdomide, we're hijacking the native cellular machinery to do the heavy lifting for our drug molecule, and doing so with compact, small drug-like molecules. And like heterobifunctional molecules like this AR PROTAC, we are binding to two different biomolecules. On one side of our molecule is binding to the ribosome interior. The other part is making binding contacts with that linear peptide chain. The difference in this case is that we don't need to induce proximity. It's guaranteed. The peptide chain has to go through there. And we don't need to bring things together. The space is already very compact and tight. It's just about creating that selective friction in between. So we have two main use cases that we've pursued so far, but of course there will be many others in the future. One such use case is in the space of oncology, where very short half-life oncogenic drivers are emerging as targets of high interest to us. Here I'm showing on a log scale a distribution of half-lives across the entire proteome. And the median half-life in HeLa cells is about 40 hours. In B cells, it's closer to 90 hours. And what you can see here is that MYC, the oncogenic transcription factor, has a half-life of about 30 minutes. So really an extreme outlier to the rest of the proteome. And this is something that we exploit, and I'll show you how in just a minute. The other place that I alluded to before are these long repeat-driven proteinopathies that are the hallmarks of certain neurodegenerative diseases like Huntington's and ALS, where, for example, in Huntington's, a long poly-Q expansion is a very unique sequence that is not well represented in the rest of the proteome, and is something that we can really address. So coming back to the oncology setting, so again, these short half-life targets have a high translational demand. They're being synthesized at a very, very fast rate, but they're also clearing at a very fast rate from the cell, as shown with the metaphor of this bucket here. Whereas the rest of the proteome is more on a drip feed in and a slowly draining pool of the target in the cell. So in effect, when we turn off the tap to new synthesis by blocking the ribosome with our molecule, we see that bucket go dry in the case of these short or very fast turnover targets. And a cell that's addicted to MYC as a transcription factor can't survive without MYC for more than a couple of hours before it goes down the path immediately to apoptosis. Whereas if we have a partial slowing of a few other targets during the action of our molecule, we may slow the rate of translation a little bit, but because the pool is draining very slowly, we would have no appreciable phenotype. And we demonstrate this here with this tumor cell growth figure that I'll walk you through. So there are two different cell lines here that we're looking at. In red is the MYC-dependent MCF7 breast cancer cell line, and in black is just a normal HEC293 epithelial kidney cell. And what we're trying to do here is, understanding that in a typical cell viability assay, it's not really representative of the clinical situation because it's a fixed system, a closed system with a fixed concentration of the drug molecule in there for the full 72 or longer hours, where when one takes an oral medication, there's a Cmax, and you have draining of or the clearing of the molecule over the course of the dosing cycle. So we try to simulate that more closely with washouts here. So we treat the cells for 6 hours with the drug, and then we wash out for 18 and repeat that each day. And what you can see is even that only with that 6-hour exposure of the drug molecule, that's enough to completely curtail the growth of these MCF7 cancer cells where we have no appreciable impact on the growth of the normal epithelial kidney cells. And so this is leveraging the kinetic aspect of this modality also for maximum efficacy and safety. So MYC, as I alluded to, is one of the most sought after drug targets, but has not been drugged yet. And even though it's responsible for close to 90% of all cancers, including those that have MYC amplification, what we see is that MYC is undruggable because it's intrinsically disordered. So even finding a binder to MYC has been particularly challenging. But I would argue that the bigger problem isn't so much finding a binder; it's just this very fast turnover. So any kind of a occupancy-based approach would be immediately overwhelmed by a continual influx of newly synthesized MYC into the cell, and you can just never keep up from a PK standpoint. Again, our molecules show broad efficacy in cells that are almost all entirely MYC dependent across this panel here, from a potency of 10 nanomolar to 100 nanomolar in this set that represents different solid and heme onc tissue types. We can show by Western that our molecules can induce the clearance of the target of interest by simply turning off new synthesis. And we show this by Western, where we are getting depletion of MYC in a cancer cell by in a very rapid time course that's consistent with what we know about MYC's turnover kinetics. And we're pleased to see also that it's affecting downstream target cyclin D, which is a MYC-regulated gene. And we can also confirm this with the orthogonal technique of HTRF. And this, of course, then translates into potent cell killing in multiple different cancer cell lines. Here I'm just showing 3: prostate cancer, a hormone-positive breast cancer, and a triple-negative breast cancer line. But again, very similar potencies. When we go in vivo, we have even more potent effect. And here is a triple-negative breast cancer xenograft study that was done, where we were at a very low oral dose, 1 or 2 milligrams per kilogram oral once a day. We're getting very robust tumor growth inhibition along with extremely well tolerated behavior in terms of body weight, et cetera. This is a very difficult to treat triple negative breast cancer cell line. It's the clinical options are really only surgery, radiation, chemotherapy. Of course, more recently, pembrolizumab has been approved for triple negative, but again, a very difficult to treat line. We can follow up after this study and look at, in the tumor sample to show that we are indeed depleting MYC levels significantly, and that is also resulting in a depletion of cyclin D levels. One of the things that is in the back of our minds is because of experience with the GSPT1 degraders, where the dose-limiting toxicity seen in the clinical setting is thrombocytopenia and neutropenia. So we deliberately looked at a complete blood count panel at the end of our study here to show that there were absolutely zero impact on any blood cell types, again, further underscoring the safety and tolerability of this mechanism. We can go back in the same triple negative breast cancer line and push the dose even more. Here I'm showing 3 mg per kg, almost flatlining. And then at 6 mg per kg, we're basically on top of chemotherapy in the form of paclitaxel. Again, very well tolerated from a body weight standpoint. We have multiple other xenograft studies in different tumor lines that we've accrued over the last couple of years. And we have further preclinical models in patient-derived xenografts that we're doing in partnership with an academic investigator at the Fred Hutch. So one of the hallmarks of this modality as well in the cancer setting is that we have a bimodal mechanism of action based on blocking translation. So the CDK4 inhibitors like palbociclib show that they are basically inducing cell cycle arrest, whereas Taxol is inducing apoptosis. We have at lower concentrations induce cell cycle arrest, but at higher concentrations show induction of caspases and apoptosis. So we're getting a big bang for the buck close to Cmax in the animal and will be in the human to make irreversible changes to the growth of those tumor cells and induce cell killing as a really potent mechanism. Again, I told you about our progress there in oncology. I want to switch gears a little bit and talk about some of the applications in the neurodegeneration space, and in particular in the long repeat-driven proteinopathies. So we're emboldened by the recent successes that in neurodegeneration space, that a very difficult and challenging part of drug discovery, but with antisense oligonucleotides showing both in the case of Huntington's disease that driving down the levels of mutant Huntington can show a slowing of caudate atrophy. And in the case of Alzheimer's disease, that knockdown of tau by ASO, by the Biogen molecule, shows that you can actually begin to reverse the pathophysiology of the disease by blocking new synthesis with an ASO. You can begin to see some disaggregation of the aggregates that have accumulated. And this is really exciting to us because unlike in the oncology setting where our goal is to drive the level of the target down to zero by blocking translation, in the case of neurodegeneration, we believe that this demonstrates that even a 50% knockdown can be really game-changing. So we have three different areas that we are focusing on. One is the repeat-driven proteinopathies, where these unique sequences represent a really interesting physical-chemical environment for our molecules by this modality. We also are pursuing, in potential partnership with a large pharma company, some tau and synucleinopathies. And then, of course, we can, with our interdictionary, look at across other target classes for things that are a good match for our chemical matter. So if we turn to these polyQ expansions and the C9orf72 proline arginine and glycine arginine duplex repeats, these again are very unique signatures for sequence that aren't represented well in the rest of the proteome. So we can really optimize molecules on this basis very readily. And the thought is that, you know, even as a single amino acid that our molecule may have a very, very high affinity for is like a little speed bump. And in the case of a single speed bump, you may experience a minor slowing of the translation, but a resumption to rapid translation after just that tiny little hiccup. But if you have speed bump after speed bump after speed bump of all sequential iterative interactions with the same amino acid, those accumulate to a threshold where then again those quality control mechanisms kick in for the cell to abort the translation. And of course, because these polyQ expansions in the case of Huntington's are also represented in some of the spinocerebellar ataxias, we could potentially have a single drug that could address multiple diseases. Again, coming back, the ASOs showing promise there, but splicing modulators as well, with both PTC and Skyhawk recently showing data that by blocking inducing improper splicing, you can interrupt the mutant Huntington synthesis And these have shown, successful knockdown in the clinic, but it's yet to be determined whether there'll be the ability to discriminate between the wild type Huntington, that is a shorter polyQ, versus the mutant and pathogenic form. And that's where our modality has some really great advantages. Clearly, the interaction of glutamine is both a feature that we can intervene on with our molecules, but it's also the same component forces that induce the aggregation of these neurotoxic proteins. It's the same physical chemical donor acceptor features that are kind of unique to glutamine as an amino acid that are the basis of forming these aggregates. So what we can show is by a hybrid assay that we've developed, that we can show a much better synergistic effect with a longer glutamine stretch than with a shorter one. So here I'm showing ratios of IC50s for the speed of translation on a polyQ stretch of 144, which is representative of the pathogenic form, versus a queue length of only 24. And so obviously, because in order to length match, the 144 is 6 times as long as the 24. When you're getting 15-fold difference in the rate of translation, it's beyond just the mathematics of the length match. It's actually showing evidence of this synergy of, again, repeat insult of a minor slowing on a glutamine after glutamine after glutamine as this ribosome is reading along this mRNA, and the molecules interacting with the peptide. Those add up to a sufficient slowing to cause ribosome collisions. Here we also have a nice cryo-electron microscopy structure of one of the molecules that was designed against the polyglutamines, and showing the very strong both shape complementarity and electrostatic complementarity. And we can leverage these for iterative additional rounds of structure-based design as we progress this program. So with that, I just wanted to thank again, amazing team that have been tirelessly working towards making this a reality over the last several years. It's been the most exciting science and the most amazing team I've had the pleasure of working with in my career, and excited at what we discover every day. And we have a bunch of additional advisors and consultants that have been also extremely helpful for as we bring this modality forward. And I want to thank everybody for their time and attention in the opportunity to present here with Drug Hunter today.
Awesome. Yeah. Thank you so much, Larry. That was a great summary. Really learn. Or you want to say something about that last slide?
Oh, no, it's just, yeah, here, if people want to read more, there's our bioarchive paper, and please visit our website as well.
Yeah. No, really amazing how you can, target those proteins before they get built even. That's a very much ideal and novel idea, even expanding on the idea of protein degradation, targeting the protein before it even gets formed. So super fascinating. We have a few questions by the audience already, but I actually have a few questions of my own too. Maybe we can start with one question. I was wondering, you highlighted very nicely the short half-life of MYC and how this is, very essential to the mechanism of the interdictor. And that probably proteins with kind of a shorter or longer half-life are probably not that prone to that type of mechanism. That makes me think, like, what are some other like interesting targets that have been hard to drug by traditional methods that you can potentially drug with interdictors that have that short half-life?
Yeah, thanks, Dennis. So obviously, some of the targets that we're pursuing in that space are proprietary, but what I
Yeah.
can say is that we've done a pretty comprehensive analysis across the proteome, and invariably, those targets which have that very short half-life are, generally speaking, transcription factors and oncogenes, where they're only needed for during certain parts of cell cycle. And
Mhm.
so those represent very nice places to intervene because again, we don't do anything to the existing pool of the target. We're merely shutting off new synthesis and waiting for that pool to drain. So things that drain very quickly are then going to be gone quickly, and we can leverage that pulsatile dosing kind of mentality to actually get the therapeutic benefit.
Yeah, that makes total sense. Yeah, very excited to hear about more target when they become public. So John Overington is wondering, do your ribosome binding compounds primarily bind to protein or RNA composed binding sites?
So they bind to the interior of the ribosome, which is ribosomal RNA. In fact, 90% of the RNA in our bodies is ribosomal RNA as opposed to messenger RNA. So we don't make any direct contacts with the messenger RNA, just the ribosome architecture and then that linear peptide chain.
Yeah. Is there kind of a common motive that can be used for ribosome binding? We're talking about, heterobifunctional, and of course you're very nicely targeting selectively the growing amino acid chain. I was wondering if they're similar to kind of protein degraders with Serblon, for example, if there's a common structural motif that's binding the ribosome, or if it's a little bit more differentiated in that case.
No, that's another wonderful question. Yes. So we can essentially, we have a core molecule that can be applied across every different target, and we can certainly just change moieties that are making the contacts with the peptide chain. But there are cases where, we can also tip the balance of the binding energy to be more weighted towards the ribosome or more weighted towards the peptide chain, depending upon the particular context that we're interested in, and we can make modifications to affect that relative contribution of binding energy from those two relative pieces. And there's also places where we can move, grow our molecule to extend either deeper or higher up in the peptidyl transferase tunnel, depending upon the effects that we want to get. And so some of this is still part of our learning. But again, the academic community has also been studying a number of different molecules in this regard for years as well, where we can retrospectively get some additional clues.
Yeah, amazing. Cool. We have another audience question here wondering about the inner dictionary. Does your inner dictionary predict off-target protein synthesis? And do you do proteomic-wide scans to see if other protein synthesis is affected?
So we do leverage proteomics. I think one thing that's important to understand about the application of proteomics in this modality versus, say, a degrader context is, again, we don't do anything to induce the clearance of the protein. We're just turning off new synthesis. And so if we were to do a proteomics experiment, the time course of that in order to be able to understand the range of everything that might be affected would have to be tied to the half-life of that given target. And so again, if we block synthesis of something that has a 100-hour half-life, if we block new synthesis for a couple of hours, you're never going to see a difference in that level. And so it really has to do with understanding the time course that's relevant to the PK properties of your molecule and the importance of those targets. Not every target is an essential gene. Some things are only synthesized under certain conditions. And so it's a complex undertaking to really understand the global changes, just as it is with cereblon or other targeted protein degraders, to really understand the scope of changes.
Yeah, absolutely. Thank you. Andy Tsai is wondering about how selective would these interdictors be for particular protein sequences? It looks like they can be selective for 5 mers. So, probably the question is about, can you get larger amino acid sequences larger than 5 mers?
Mhm. So yeah, 5 mers is about the amount, the length of peptide sequence that the molecule can bind at any given snapshot in time. But remember, this is a moving train, and so a new peptide bond is being formed in the ribosome every 300 milliseconds. So at any given time, we're binding to 5. If we look at MYC as an example, MYC is 454 residues long. In MYC, there are 20 5-mers that are in no other target in the entire proteome. There are 143 5-mers that are in 5 or fewer targets in the rest of the proteome. So the scarcity of these sequences is a lot more than you would imagine. It also matters, though, not just the sequence of where that 5-mer has the strongest interaction across the entire length of the protein, but it's what comes before it and what comes after it. So that same 5-mer that would be flanked by prolines that are naturally slow translating is going to have a compounding effect and a much different result than that 5-mer in a different target isolated without those prolines nearby. So these are all part of the learnings that we leverage from the interdictionary to understand not just the sequence preferences, but the location and the context of the sequences.
Yeah, yeah, great. Amazing. Yeah, following up, Yoshi is wondering, following up on this earlier question about selectivity, how do you check for selectivity across other proteins in other cell lines, or is the selectivity not as important? Because, for example, MYC has this especially short half-life. You showed that with the kind of bucket analogy, right?
Right. So yeah, exactly. It's kind of context dependent. So in the case of we have to consider the exposure profile of the molecule, whether it's in a cell or in an animal or a human, to be part of the equation. And then again, it's also depends on, certainly in the oncology setting, our goal is to, again, to drive the level of the disease-causing protein down as low as possible to have the maximum effect on cell killing. But in the neurodegeneration case I showed, and in potentially in immunology, where we're looking to dampen things down to a place far less than 100% knockdown for therapeutic benefit, then we can be a lot more gentle in the how much dosing we need to do, et cetera. So it's really it's very context dependent.
Yeah, yeah, makes total sense. And then John is wondering about the mutation rates in ribosomal RNA in cancers. Is there anything known about this? As you said, 90% of the cancers are MYC driven. Particularly in those cancers, anything known about the ribosomal RNA mutations?
Yeah, so the ribosome is really would be difficult to mutate any to any appreciable degree without catastrophic consequences across the entire organism. And so for that very reason, we believe that this modality will have an extremely high barrier to resistance. And as this is one of the virtuous features of this, that we should be able to have a long runway of efficacy in a clinical setting without resistance emerging. And of course, then that means that it's also would be an ideal combination with any other oncology agents. But the ribosome mutations in the ribosome are not easy to do without significant consequences.
Yeah, Yeah, that makes total sense. And then Curtis Tyree is wondering, you showed, I think, the hematological toxicity data, and that there's essentially no concerns there, essentially, with that toxicity. They're wondering if you see those effects on the blood cells when you push up the dose, or if they're not seen at all.
We've not yet seen that. We're looking at other orthogonal means to understand the potential impact on myeloid progenitor cells as one of several rapidly proliferating cells with a higher translational demand. But so far, we have not seen that.
Yeah, that's great. That's great. And then we have another question from Yaguang C. Um, they're wondering if IDB 1928 blocks MYC translation in mouse also. I'm not sure. Is mouse MYC different than human MYC? I'm actually not sure.
Yeah, no, the first of all, the ribosome is extremely highly conserved across species, and a single amino acid change is unlikely to confer insensitivity to our mechanism, again, because of just the dynamic nature of that peptide chain going through. A single amino acid being different isn't going to have a huge impact on overall sequence.
Hmm. Yeah, and then I guess another question here about mutations from Stuart Huang. Can you comment on the emergence of adaptive mutations or pathways to the IDB treatments?
Um, Sorry, could you repeat the question?
Yeah. Can you comment on the emergence of adaptive mutations or pathways to the IDB treatments?
Yeah, so far we haven't been able to see any, adaptive mutations, but we haven't, again, that's an undertaking that would take a long time of exposing cells and seeing if there were, again, we're highly doubtful that there could be mutations in the ribosome, and we don't expect point mutations in targets of interest to have any impact either. So, but it's not something that we've been able to comprehensively address yet.
Yeah, sounds good. And then, yeah, we have another question here. Down regulations for MYC seem somehow limited, especially in the in vivo data. Do you see this as a potential issue, or to achieve a maximum therapeutic index for different targets and diseases?
Yeah, one of the challenges that is the magnitude of the suppression of MYC in the tumor sample is partly reflecting just the technical challenge of because MYC is being turned over so rapidly that in the time that it takes the technician to harvest the tumor and flash freeze it, there are already changes happening. And so this is a limitation of when you're dealing with a target that has a 30-minute half-life. It's and a rapid resynthesis rate, and trying to work quickly to harvest a tissue sample. It's just partly reflecting that.
Makes sense. Yeah, I think yeah, we have some more question, but probably in the interest of time, we would need to wrap up now, Larry. So I want to thank you again for an amazing presentation and sticking around also to answer many of our audience questions today. Really, I learned a lot today about the MYC half-life, about those neurotoxic peptides that you showed in the end of the talk. Really fascinating, and I think a very promising modality. So again, thank you, Larry, for introducing us to the interdictors and sticking around to answer all the audience questions.
Thank you, Dennis. It was a pleasure to participate. It really a fun experience. Thanks.
Awesome. And yeah, we will be back here on October 2nd. We'll have Fabio Broccatelli talking about From Molecular Properties to Mechanistic ATME, a guide to scoring success. And I hope, of course, to see everybody there at that particular Flash Talk. But until then, I wish everybody a wonderful rest of your week and a wonderful weekend. And we will see you in early October. Thanks again, Larry.
Thank
And
you.
bye, everyone.
Bye.
Bye-bye.